Hello, everyone, and welcome. I'm Jack Van Horn from the University of Virginia. I'm very proud to welcome you to this edition of our 2023-2024 Foundations of Biomedical Data Science Seminar Series. The lecture series is supported by UVA's Integrated Translational Health Institute of Virginia, the UVA College of Arts and Sciences, the UVA School of Data Science, and through a grant from the National Institutes of General Medical Sciences. Since 2016, the Foundations of Biomedical Data Science Seminar Series has focused on the use of data science methodologies, artificial intelligence, deep learning, and statistical modeling as they pertain to different themes in the biomedical and health sciences. Our theme this year is focused on building partnerships for generative AI training in biomedical and clinical research, where every Friday we enjoy presentations from a wide range of leading thinkers about the issues, promise, opportunities, and hurdles associated with the emerging trend in AI-based methods. Indeed, we'd like to remind folks that our 2023-2024 application cycle is still open for another a few hours, and we encourage junior faculty members working in areas such as the development of generative AI-based methods applied to biomedical research, and those junior faculty actively utilizing AI in their biomedical teaching to consider applying. We believe this is an exciting and engaging program, which this year will bring together AI platform developers and university-level educators to better understand the power, the promise, and the potential pitfalls associated with the generative AI explosion. Participants selected for our Biomedical Data Science Innovation Lab program will leverage these seminar presentations as vital material for our culminating in-person manuscript and grant project development workshop to be held at the lovely Bahia Beachfront Resort located in sunny San Diego, California in June of 2024. And today I am delighted to welcome our speaker, Dr. James Zhou, from Stanford University. James is an assistant professor of biomedical data science, computer science, and electrical engineering at Stanford University. He is also the faculty director of the Stanford AI for Health program, and he works in both improving the foundations of machine learning by making models more trustworthy, reliable, as well as in-depth scientific and clinical applications. Many of his innovations are widely utilized in technology and the biotechnology industries, and he has received a Sloan Foundation Fellowship, an NSF Career Award, two Chan Zuckerberg Investigator Awards, and a Top 10 Clinical Achievement Award, several Best Papers Awards, and a whole bunch of other awards as well. And in continuing with our 2023-2024 Biomedical Data Science Seminar Series theme, James's talk today is going to be entitled Biomedicine in the Age of Generative AI. And James is going to note that there have been tremendous advances in generative AI in the context of ChatGPT, DALL-E, and various other generative AI systems. And his talk is going to explore how these models can power biomedical discoveries by expanding the design space of medicine while balancing complex trade-offs. And he'll illustrate several examples, and then he'll apply a similar generative AI approach to guide the design of clinical trials and show us how that can be done. As always, we are streaming this lecture live and for recording via YouTube. And if you are watching on YouTube, thank you so much for joining us. Also, our specially selected 2023-2024 Biomedical Data Science Innovation Lab participants and alumni are encouraged to submit any questions they have via the chat feature in their YouTube sessions. I will synthesize these questions and I will ask them on your behalf during the last 10 minutes or so of James's lecture. And James, with that, welcome. We are so excited to be hearing from you, and we're really looking forward to your presentation.
All right, great. Well, thank you very much for the introduction and for organizing this, Jack, and really nice to see everyone. So I'm James Zhou, and I'm excited to discuss and share with some of you some of the recent developments in developing and applying generative AI approaches across different biomedical applications. All right, so we've seen really exciting advances in generative AI, especially over the last year and a half, right? So for example, in the area of art, right? So people now have these great tools for typing in some descriptions of images and then having the models to generate images that they want to see, right? In another area of generative AI, that's tremendous advances, right? So people have been using models like ChatGPT in your everyday lives. And in addition to ChatGPT, now there's a whole ecosystem of large language models, and there are new models that are coming out, on every week or every couple of weeks. And these kinds of models and generative AI tools have also been very useful in biomedical research and education, right? So for example, I was teaching a class recently at Stanford for students, right, looking into computational biology, right? And as a part of a class, right, so we actually help the students basically use tools like GPT-4 to help them to generate analysis code, right? And with the help of GPT-4, the students were able to actually replicate some of the quite complex analysis that were published in some of the original pioneering papers, but now using the help of GPT-4, they were able to replicate this analysis in a much shorter amount of time, right? This also gives them more time to look into the more in-depth outcomes and interpretations of the data. Right. So what I want to discuss today, right, is really some of the thoughts on like what really makes generative AI different, and how do we really leverage these tools for biomedical applications. And if we first take a step back, right, so like how is generative AI actually different from other kinds of AI or machine learning systems? Right. So if you think about the more standard statistical machine learning models, right, these models are usually meant to make predictions, right, regression problems or classification problems. And in the setup of these models, right, so you have some inputs, the model could still be a pretty complicated multi-layer neural network or your favorite machine learning models. But typically, the output of the model is usually relatively structured or relatively simple, right? If I'm making a regression problem, let's say I'm trying to predict the survival outcome of a patient, right? Usually the output will be just a single number corresponding to survival time. If I'm making like a classification problem, the outcome then could be maybe one of a few classes that are fixed ahead of time. Right. In contrast, right, if I look at the recent generative models, right, the outcomes, the outputs of these models are typically much richer and much more complex. Right? Instead of having these a few categories or a few scalar numbers, right, the output of these generative models could be images, like we saw on the first slide. It could be molecules, which I'll explain next. It could be code, right, analysis code or software itself. And the models themselves can also be substantially larger than the models that we used before in terms of the number of parameters. Right. So from that perspective, then, right, it's really the way I think about a generative AI is that it's less about making specific predictions, but it's more about actually designing, right? It's helping us to explore this more complex space of of images, of molecules, of code, of algorithms, right, and help us to really design these outputs, design these complex objects. And today I want to explore this idea of design, right, enabled by generative models. And in particular, we want to see how does these AI generated designs can help to facilitate biomedical applications and translations. And I think biomedical translation pipeline is an especially good application area, right, for generative AI, because typically when we think about biomedical translation, we have to balance these quite complex trade-offs, right? Trade-offs like the cost of experiments, right? The efficacy of the experiment, the safety to patients. And when we think about this entire pipeline, right, all the way from early stage discovery, right, we're doing drug discovery or target discovery, to later stages of clinical trials, to all the way to post-clinical settings, right, at every step, they involve some complex designs to balance these multidimensional objectives. So today I want to give some concrete examples of how we can actually use generative AI to help us to improve our designs across this entire pipeline, right? From early stages, right? I'll show you an example of how we actually use generative AI to help us to explore the space of new molecules, to design new drugs. And in the middle, right, after we have these new drugs, we need to design clinical trials, right, to effectively evaluate these drugs. So I'll also explore how we can use these generative approaches to help us to design more inclusive and more diverse clinical trials. And even after the drugs come on the market, right, there's still questions around how do we assist physicians, clinicians, try to figure out what drugs to use, or how to diagnose patients, and what drugs to use for a given patient. So the last part I will explore is how do we use generative models to basically provide clinical assistant tools for clinicians and researchers. Okay, so this will be the three parts of the presentation today. So we'll dive in by looking at the first part, right, which is thinking about how to use generative AI models for designing new drugs. And I want to give you a concrete example of how to use these tools to design new antibiotics. The kind of algorithms I'll describe will be quite general, so they can be used to do other kinds of small molecule drug discovery. But I want to give a concrete example of antibiotics because it's, I think, really an urgent and high impact problem. All right, so many of you know that antibiotics was discovered in the 20s with penicillin. And what's interesting is that if you look at the history of antibiotics, right, the first few decades after penicillin, there's actually a sort of a gold rush where there's a lot of new antibiotics, new molecules being discovered and put on the market. But it's important to know that basically for the last 60 years, there are actually relatively few structurally novel antibiotics that people were discovering and then bring into the market. And this is a problem, right? Because if we don't have a lot of different diverse kinds of antibiotics, then it becomes easier for bacteria to develop resistance to the existing antibiotics, right? And the fact that bacteria have developed resistance to many of the existing antibiotics is currently a major public health crisis, right? So currently over 1 million people die every year because they're exposed to bacteria that have developed resistance. And that number is unfortunately expected to really increase quite a bit over the next few years, right? And this is illustrated by this timeline here that's shown here in the bottom, right? Where above the timeline indicates when the first time when a new antibiotic drug is first introduced, brought into the market. And the bottom corresponds to when we first observe resistance, right, to that drug. And the main takeaway here is that in many of these cases, right, there's only a really relatively short lag between the introduction of the drug and when resistance develops, which really highlights the fact that we have this ferocious arms race, right, between drug developers and pathogens. And because we're not discovering as many new novel antibiotics, right, so we're really losing the risk of losing this arms race. As a consequence, now we have these bugs, like this one called here, Acinetobacter baumannii. It doesn't look very dangerous, but it's actually an extremely dangerous pathogen. It's considered by experts to be the perfect predator because it has developed resistance to many of the existing drugs that we have. Right. So in this first example I want to give is I want to describe how we've actually used generative AI to help us to discover new novel small molecules, right, that have then we've experimentally validated to be effective in killing this dangerous pathogen. Okay, so here's where generative AI comes in, right? So the idea here is that we want to use these AI models, right, to help us to explore the vast space of small molecules. Right? So small molecules are typically the most common kinds of drugs that we have. And many of them have been tested and been synthesized. But if we look at the actual space of potential small molecules, right? So we have only ever experimentally tested a very small sliver of all the possible small molecules. And this is where I think AI can be very helpful, is to help us to explore this vast space of the universe of small molecules, especially of novel molecules that we haven't actually seen before or experimentally tested before, and help us to decide and create new molecules that then we can synthesize and test. Right, so I have a fantastic PhD student, Kyle Swanson, who's a student working with me at Stanford, and he developed this really nice pipeline, which I'll describe next, for using generative AI approaches, including some reinforcement learning approaches for developing and discovering these new small molecules. But first, I think the big challenge, right, in using generative AI for drug discovery is the following, right? It's actually relatively easy for generative AI models to generate hypothetical molecules, right? They can hallucinate new molecules, but the hard part is we don't know how to make these molecules, right? It's easy for the algorithm to come up with some new molecules that nobody has seen before, right? But if we don't know how to make those molecules, then that doesn't really help. It doesn't really advance drug discovery. So then what we want to do in Kyle's algorithm is that we don't want to just have the AI model to create new molecules, but we want to ask the generative AI model to help to generate new molecules and also the recipes to explicitly make each of these new molecules, right? So here's one of what we mean by a recipe for synthesizing new molecules, right? So it's sort of like a cooking recipe, right? So you start with building blocks, which are sort of small fragments of molecules that are easy to create, and we know how to make these building blocks. And then we can apply chemical reactions. There's a list of well-known chemical reactions. Each reaction, you can think of this as basically being like a template that can be applied to a subset of these building blocks, right? So in this reaction, I apply it to these two building blocks. And then the output of the reaction will be like a larger, more complex molecules. Right? And you can also apply a sequence of different reactions to create even more complex molecules. Right? So this is sort of like the space of potential molecules that we can potentially synthesize and explore. And it's a huge space, right? Because we have on the orders of hundreds of thousands of fragments. And there are also maybe hundreds of reactions that people know how to work with. And if you apply these in different combinations, you can quickly see that we have this really exponentially large space of potential molecules. And in the space of potential molecules, right, it's worth emphasizing that people have only ever tested and built, constructed only a very small subset of the potential molecules. So this is where the AI comes in, right? So we want to basically use the AI model to help us to create these recipes, where each recipe then will be a step-by-step set of instructions of taking, for example, different building blocks, apply reaction 1, and then apply reaction 2 to create a new molecule. So the way that we model this through the AI algorithm is through this basically what we call like a multicolor tree search. As this is inspired by some of the recent advances in AI that's used to play games, right? Like, for example, there's a famous AI algorithm called AlphaGo, which is very good at playing Go, and it's also a similar algorithm for playing chess. And the way that these game playing AI algorithms work is that they basically model the game as a huge space, a tree of potential moves, right? And then use the AI to sort of explore that space of potential moves. So in our setting, we also have this huge tree, right? Where instead of the nodes being the potential moves that you can make in chess or Go, the nodes now corresponds to different molecule fragments. Right, so let me explain that in a bit more detail, right? So we first start from the level 1 of the tree, and then we go down, right? So the level 1 of the tree corresponds to the building blocks, individual building blocks, right? Again, each building block are one of these small fragments that we know how to make efficiently. As I go from level 1 to level 2, right, then I have to decide, which combinations of building blocks to put together, right? Maybe in this node, now I take building block 1 and building block 2. And as I go to level 3, now I have to decide which chemical reaction to apply to this combination of building blocks, right? So here, maybe I'll apply reaction 27, and then the output of at level 3 then will be a more complex molecule, right? So you can imagine that as we keep going down this tree, right, as the further down we go, the more complex molecules we can get. And what's nice about this tree representation, right, is that each path of the tree from the root, right, to level 1, 2, and 3 to the leaf, right, corresponds to actually a very explicit recipe for how to make the more complex molecules at the leaf, right? In this case, it says that go and take building block 1, add building block 2, apply reaction 27. Is how I get this node, this leaf node. So this is sort of the representation that we want to work with. The problem here is that this is an extremely large, like an astronomically large tree that we have to search over. And this is where we can try to use AI to help us to efficiently search this space. So the way intuitively that this works is that basically to help us to guide which part of the tree to search, right, we built another AI model, which is called a property predictor. Essentially, you can view this as providing some sort of reward to say how promising it is to explore a particular part of this chemical tree. Right, so this property predictor is basically a model that we trained on a bunch of experimental data where we actually have collected drug screen data, right? And we train a machine learning model, which is like a graph neural network, to predict how, for a given new molecule, or how likely is it to have some antibiotic properties, right? So once we train this property predictor, then we use it to basically, we apply it to evaluate each of the new molecules in this tree, right? And say, okay, and these new molecules will have some sort of score, right? And a score then can be back propagated up to this path to also to provide a score for the building blocks to see how promising that building block is for leading to a useful drug. And in addition to this property predictor, right, so there's actually a bunch of other considerations that we want to encode when we try to discover new molecules for drugs, right? So efficacy is certainly one important consideration. So that's captured by this property predictor. So efficacy here means how likely is this molecule to be a good antibiotic molecule, right? We also want to have molecules that are structurally novel, right? Basically, it's different from molecules that we've seen before. And the reason why structural novelty is useful here is because if it's novel, then it's less likely for existing pathogens to have resistance to those drugs. Of course, we want the drugs to be safe for humans, right? So safety is a consideration, And also, as we said before, we want this to be very easy to synthesize, right? Basically, we it into essentially the scoring function that we applied to score each of these nodes, right? Each of the combinations. And then basically the generative AI model basically uses this scoring function as a guide to then explore different parts of the search space to look for the molecules that have the highest score, right? That has the best metrics along each of these different axes. So all of this, so this is sort of the generative AI model that we call Synthesizable, right? That's developed here at Stanford. And this is where this all developed, this is a computational algorithm that's all trained in silico. So after we've trained and built this model, right, so then the fun part comes next, right? Because now we actually get to synthesize these molecules in the lab and then really test, do they work? So to do this, we work with our actually Enamine, which is a company based in Ukraine, right? So they specialize actually in making small molecules, right? So we sent 70 recipes created by our Synthesizable AI model to Enamine. So each of these recipe then corresponds to basically a path from the root to a leaf, right? So it corresponds to basically a new small molecule and also a step-by-step set of instructions for how to make that small molecule. So all of these 70 recipes corresponds to 70 entirely novel molecules, basically that have never been synthesized before, have never been seen before on Earth. Right? And I guess the interesting thing is that because the Enamine is based in Ukraine, that you can imagine that they have a lot of other constraints over the past year, including, especially with the war going on in the background. So basically, they were not able to do a lot of optimizations of these recipes. In other words, the recipes that's produced by the AI model should be extremely simple and very robust, right? Even without too much tweaking and optimization, people should be able to just follow the recipes and make the molecules. And remarkably, it actually worked quite well, right? So Enamine was able to synthesize 58 of the 70 new molecules, right? These correspond to now like novel molecules that are potentially drug candidates for antibiotics, right? So the hit rate here would be over 80% in terms of the success for synthesizing these molecules, right? So this validated already one of the key components of our model, right, which is actually create molecules that are easy to synthesize because by providing this explicit recipe. So then we basically take these molecules, bring them back right from Ukraine, and then our collaborator, John Stokes' group, was able to validate these molecules, right? So what they do here is that we basically have 4 extremely dangerous pathogens. They're called escape pathogens. It's considered to be by the World Health Organization, WHO, to be the highest priority of pathogens because they have developed resistance against many of the existing drugs. So this includes this Acinetobacter baumannii, like this perfect predator we saw before, and also several other pathogens. And then what we did, so basically we tested each of these 58 novel AI-designed molecules against all 4 of these escape pathogens to see how effective it is the molecules are at killing these pathogens. And among these, we identified basically 6 novel antibiotic molecules that are produced by the AI. These 6 have shown to be very effective in killing all 4 of the pathogens, right? So they work quite well across the board in experiments. And we did some additional testing, right, to show that these 6 new molecules are effective, right? They're active even when they're used at low dosage. So this is important because if they're effective at low dosage, then it means that they're also less likely to lead to side effects in patients. We have mouse model studies, animal studies to show that these new drugs, they have few signs of toxicity, which is also a promising initial signal to show that they are potentially safe in humans. And we also follow, we apply these new drugs, right? We also follow them over time to see that there's actually low microbial resistance that's developed against these new molecules, which is also promising. Right, so I think all of these are very exciting experimental results that validates some of the efficacy and potential safety of these new molecules that are designed by the AI systems. Of course, this is just still an early step, right? We need to do additional clinical trials to validate these molecules in patients. But I think this is already showing a lot of promise for seeing how we can use generative AI to help us to explore, right, entirely new spaces of small molecules, and also to produce new molecules and also instructions for how to make these new molecules, right, that are easy to follow and likely to be effective. Okay, so this is the first example of in the early stages of the biomedical research and discovery pipeline, right? How we can actually use generative AI to help us to explore new designs and in this case, new drugs. So now that imagine that you have to produce some new drugs, right? So typically the next step in this pipeline is you want to start to design clinical trials to test these new drugs. Right. So for the next vignette I want to give is I want to demonstrate how we actually use these AI algorithms to help us to generate designs of clinical trials, right? With the goal of making clinical trials both more efficient and also more inclusive. So clinical trials is really the bottleneck and also the gatekeeper of medical research and medicine, right? So you can start with hundreds of great ideas promising new drug candidates, and they have to go through different phases of clinical trials to test for efficacy and safety. And only one of these 100 great candidates can actually make it to the patients, right? Because many of them actually failed the different clinical trials. Right? And because each trial itself can cost on the orders of tens, sometimes hundreds of millions of dollars, right? So then accounting for all of these failed trials, right, the average cost of bringing a drug to patients then is several billions of dollars, right? So then anything that we can do to make the trials, right, more effective and also more efficient, then can be really broadly impactful for the entire biomedical translation pipeline. So that's motivated us to see how we can use generative AI to help to improve the designs of clinical trials. And I want to describe to you one platform that we built called Trial Pathfinders that was described in this paper that we published in Nature. And this Trial Pathfinder, I'm excited to say that it's actually now also being used by several pharma and biotech companies to help to guide the designs of their new cancer trials. This is work that's led by Rishan Liu, who was an excellent PhD student working with me at Stanford. So here's the setup of the problem, right? Basically, we want to use AI to help us to decide which patients to enroll into the clinical trials. So why do we want to look at this patient enrollment, right? So the clinical trials is actually very complex because they are extremely selective in what patients enroll. Right? So let's say if you look at a trial that look at a particular disease, maybe lung cancer, right? So among all the patients that have that disease, only a very small fraction of these patients are actually eligible to participate in the clinical trials under the current designs of these trials, right? So for example, each clinical trial would have pages, right, of criteria to filter out patients. Some of the patients might be filtered out because they have different comorbidities, or maybe some of their lab values are outside of the range, right? Or because they have different previous conditions or different subtypes, right? So these patients are all filtered out. So the remaining patients are often called by the industry called these Olympic athletes of the patients, right? So many of these trials are looking for these Olympic athletes of the patients because they're thought to be like the safest patients to enroll in these trials. But this overly restrictive filtering of patients is a huge barrier, right? So it means that many of the patients who actually want to participate in clinical trials to get access to the latest treatments, they're not eligible, right? So it hurts those patients. Actually also hurts the drug developers because a large number of clinical trials have face a big challenge in enrolling enough patients, right? And also means that because these trials are enrolling these Olympic athletes of the patients, how well the treatments work on these Olympic athletes of patients may not reflect how well the treatments work on the everyday patients like us. So there's a huge distribution shift, right? So for all of these reasons, right, people are increasingly recognizing and calling for clinical trials to be more inclusive and more diverse. And the big challenge, right, in designing clinical trials is that the current way that we design these trials are often pretty anecdotal, right? So here's just an example, right, where the details are not very important, but each row here corresponds to a particular eligibility rule that's used to filter out patients. Right, so basically, the first one just says that if a particular lab value called a neutrophil count is outside of some range, then those patients are not eligible. So I actually took a screenshot of this, from a trial that's being designed in progress. What's interesting is that you can actually see some comments in the Google Doc, right, for designing this trial, where somebody actually suggested, can we actually relax that threshold to enroll more patients, right? And then a third person says that, well, we don't really have evidence to decide how much to relax it by, right? So that's then just use the more conservative, more strict thresholds that was used before. So you can see that there's actually a lot of interest, right, in changing the different designs of the trial to enroll more patients, right? But often the big challenge is that we don't really have a great data-driven way to help us decide how much to set these different thresholds. And this is really a huge problem across the board, right? So for example, the US National Cancer Institute has a mandate, right, that all of the cancer clinical trials really should be simplified to enroll more patients. But how to really design this eligibility criteria, right, for these trials, that's still a big open challenge. And this is where we think sort of data science and generative AI could be helpful. So this is what motivated us to develop this trial Pathfinder AI approach. Right? So here's really the high-level idea of how this approach works. Basically, we're going to use AI algorithm, right, to generate clinical trials designs, right? This AI algorithm is going to be using real-world data, so basically EHR data of large number of patients to computationally emulate clinical trials. And it's going to use those emulations to help to guide the designs of new trials, right? And for this emulation, we're going to use, we use this large data sets collected by the Flatiron Network, which contains data from over 280 cancer hospitals, clinics in the US, over 2 million patients. And so it's a really nice longitudinal data set of all these different patients. So here's an illustration of how this AI generation of clinical trial design works, right? So the goal of this is to basically have the AI algorithm to basically generate or propose trial eligibility rules. Right? Like we talked about before, you can think of the trial eligibility rules as basically a set of criteria to decide which patients are eligible to participate or not eligible to participate in a given clinical trial. So first thing we can do is to basically start with the existing clinical trials, right, and have the AI algorithm to start with that as the starting point, right? So each of these eligibility rules corresponds to like a mathematical constraint. You can think of that as a mathematical constraint to filter out patients. And there could be dozens of these eligibility rules. And then what our AI algorithm does is that it's going to take a particular set of proposed eligibility rules for a clinical trial, right? And then goes into the medical records of these millions of cancer patients from hundreds of hospitals. And then it basically creates a digital cohort of patients, right, that's taking the relevant treatment and also satisfies all of these eligibility rules. And also creates like a comparison cohort of patients that's taking the same treatment, but may not satisfy all of the rules. Right? Maybe they only they miss some of these eligibility rules, right? But they're taking the same treatment, right? And because we have the longitudinal EHR data of all of these patients, then the algorithm is basically going to follow and look at the outcomes, like the safety and the survival outcomes of these two groups of patients, and see is there actually a difference, right? If there's a difference, that means that maybe there is some differences that can be attributed to the specific eligibility rules. Right? If there's less of difference, right, that means that we can potentially relax some of the rules to enroll more patients. So that's the high-level intuition, right? So then the algorithm looks at the simulation results of the trial, and then goes back to see, okay, so how can it change some of the eligibility rules, maybe by relaxing some of the, what happens if it relaxes some of the eligibility rules, right? So it proposes like a new design of the trial, and then it goes back to the EHR data to emulate this new design, right? So now, because by relaxing the eligibility rules, now there are more patients that are eligible, right? And then it can assess the outcomes. Right, so the key thing to note here is that this is all done on already collected observational data, right? So this is all done in silico, which means that it's actually relatively easy for the algorithm to generate a lot of emulation trials and then to get the data back and get the signal back quite quickly. So to summarize, right, so the trial Pathfinder is this overall approach, right? So it uses the EHR data from a large cohort of patients and then uses this AI approach to help to design, to generate new eligibility criteria, right? And it basically runs millions of these in silico trials in parallel, right? And it then aggregates all of that information together to propose a data-driven suggestion for how to design a particular trial, right? For a particular trial, right? So this is the output of the model would then we say, okay, for a given trial, then here are some of the eligibility criteria that are really important. right? And then some of the other eligibility criteria could potentially be broadened, be relaxed to enroll more patients, right? So that's the recommendation that the model provides to the clinical study teams. So we applied this algorithm, right, to a variety of different diseases, but here I just want to show you the results from applying it to advanced non-small cell lung cancer, which is one of the most prevalent forms of cancer. So here are 10 different clinical trials studying so these are phase 2 and phase 3 trials. So the last row is basically the average results across these 10 different trials. So we can mostly focus on the last row just to make things a bit simpler. So basically the first two numbers indicate that if we actually use the standard designs, the original designs of these clinical trials, here's how many patients we'll be able to enroll, right? About 1,500 patients on average for these trials. And then what is the hazard ratio for these patients, right? The hazard ratio basically measuring what is the survival outcome of patients who are taking the treatment compared to who are taking maybe standard of care, right? So lower the hazard ratio means the trial, the drug is more effective. If you want it to be lower, the better. So that's the first two rows. If we use the standard designs of the trials, the first two numbers, the last two numbers corresponds to if we actually use the AI proposed eligibility designs of these trials. We see that on average, we can more than double the number of eligible patients. So that's great. And in addition, even though the AI algorithm will be enrolling more patients, we actually see a comparable, maybe even a little bit smaller hazard ratio, right? Which suggests that even though we will be enrolling some of these less like sicker patients, right? But these sicker patients actually do respond to treatment comparably or even better than the very healthy Olympic athletes among the patients. And I think this is an important insight, right? Because the algorithm actually has learned that by enrolling, right, by relaxing some of the eligibility rules, it's able to enroll a lot more patients. And in particular, these patients then will still be able to benefit from the treatment safely. All right. And as a consequence of this, the algorithm is actually able to enable a lot more women, more minorities, and also older patients to be able to participate in these clinical trials. Right. Many of these groups are filtered out by these very overly restrictive eligibility rules, but the algorithm actually learned that we don't need to be so restrictive, right? We can still enroll a lot more of these groups of like minorities or women, but really have we start increasing the harm or increasing the side effects. So this is, I think, a really promising first step, right, of how we can actually use AI to generate, really data-driven designs. Of course, the AI is really just making suggestions, right? So this is really just additional information that the clinical study human researchers can incorporate in helping them to make the final design. So the humans are still making the final decisions of the clinical trials. But by providing this sort of data-driven guidance, this can at least provide more information, right, that can help make trials more inclusive and also more diverse. I didn't have time to go into details here, but in the paper, we actually validate these results from the AI-driven trials using data from actual clinical trials, also from other cohorts. And we have applied it to cancer, to lung cancer, but this methodology can also be applied to many of these other diseases. And we're also extremely happy that this has gotten a lot of traction in the pharma industry. It was actually selected as one of the top data science innovations by the pharma industry. Okay, great. So now if we're following our pipeline, right? So we talked about early stage discoveries and how to design clinical trials. And the last part I want to mention is how we can actually use generative AI to help us at the final stage, right? So after you have these trials, you have these drugs that come on the market, it's still a very challenging problem for clinicians, right, to diagnose patients and to decide on which drugs to give to a particular patient. And this is where we've been developing these sort of AI chatbots, right, language models that can help as an assistant to these clinicians. So I want to give one example of this, right, or where we developed AI chatbots to help pathologists. Right, so pathology is really sort of the pathologists are the people who have to help to diagnose patients, right, and they also help to inform treatment planning. And they're often looking at these images, right, taken from the tissue biopsies of the patients, and to help them with these diagnoses. And it's a quite challenging job, right, because they can often encounter images that come from different body parts, from different diseases that are very unfamiliar to them, right, they are very heterogeneous. And something really interesting happens when pathologists encounter images that are unfamiliar to them. So that many of these pathologists actually go on Twitter, they go on social network, social media, right? So here are just a few screenshots I took, right, of pathologists posting images on Twitter, right? These are high quality images that are de-identified, so they feel comfortable posting these. And then they actually invited their colleagues, right, from around the world to have a discussion of how to interpret this image. So you can see a Twitter thread of people, clinicians, pathologists from around the world chiming in interpreting and discussing each of these images. It might be surprising to some of you that your doctors are usually going on to Twitter and using Twitter to ask for help with interpreting images, but this is actually recommended practice, right? For example, the US Academy of Pathologists actually recommend that their members would use social media as a way to share knowledge, right? And we thought this is actually really great data resource for building AI models, right? Because here you have both the images and also discussions of these images, right, by the human experts, right? And this is actually great data for helping to teach like a chatbot, right, like ChatGPT type chatbot to how to interpret and help human physicians, right, in interpreting and discussing these kind of difficult images. Okay, so this is what motivated us to create this data set, so we call OpenPath. So basically, to create OpenPath data set, we first just go through all of Twitter to curate these high quality medical images along with corresponding expert discussions. All right, so to do this, we actually use specific hashtags that are often recommended used by the individual communities, right, when they're to describe their images. Right? For example, different sub areas of pathology, like renal pathology, surgical pathology, or breast pathology, have their own hashtag that their members use. Right? So that we use that hashtag to basically help us to create an initial pool, right, of millions of Twitter threads. Now, the data from Twitter can be very noisy, right? So then we did a lot of very strict filtering to remove low quality images and low quality text, right? So for example, to remove low quality images, we actually built another machine learning classifier to evaluate the image quality, right? And we use that classifier to basically remove the low quality images. And similarly, we have another classifier that looks at the text from Twitter and to remove low quality or irrelevant text, right? So there are several rounds of filtering. And after we do all this filtering, we're still left with over 200,000 high quality Twitter threads, which is currently one of the largest such public data set, right, of pathology images and discussions. And then once we have this, so basically each Twitter thread then would have like, one or more high quality images, right, that's matched with the corresponding interpretations by the clinicians. And then the next step is that now once we've curated this large data set, right, which is actually, by the way, it's all publicly available, right, so other people can also use this data set. Then we trained our own open source visual language model to help on this data to help us to interpret these images. So this is like a visual language chatbot, right? So if you think about, so basically you can give it an image and the chatbot then would be able to generate discussions around that image. Right, so the way that's trained is that we basically have like a text encoder and an image encoder. Right? And this is how many of these chatbots are trained. Right, so the idea here is that the text encoder then will look at the text information. In this case, it could be from Twitter, the tweets, right, and then extract information from the text. And the image encoder, right, would just look at the images and extract features, or we call embeddings, from each of the individual pathology images, medical images. And then the key part for building these algorithms is that we need to basically align the text understanding with the image understanding. So in order to teach the chatbot, right, how to interpret a given image. So to do that alignment is where we use the Twitter data, because we have the paired images along with the corresponding conversations and dialogues about that image, right, from each tweet. So then we use that paired information of the text and images to basically align these two encoders, and output of that is what we call the PLIP algorithm. Right, so this is basically being trained, so it's initially pre-trained on large corpus of natural images and text, and then we fine-tuned it on the pathology data that we have collected from Twitter. So here's an example of how this PLIP is used by some of our colleagues at Stanford Pathology Department. Right, so the pathologist actually could upload an image. Maybe this is the image that they would like to get some assistance in helping to interpret. Right, and then Flip chatbot would then look at this image and it will first provide some descriptions about what is in the image. Right? In this case, it says that this is a H&E image of the kidney. There's about 700 cells in the image, and there's some inflammation, necrosis, right, in this image. And then the clinician can also ask questions, right? And the model then will also provide interpretations and response to those questions based on its interpretation of the image. So in some cases, right, our pathology colleagues may not trust the generation of these chatbots because of the risks of hallucination. So to address that, we actually built a second use mode of this model, right? Instead of trying to generate its own interpretation of the image, what the model Flip does is that it actually goes to retrieve similar images that have, and discussions of similar images, either from Twitter or from textbooks or some internal like EHR databases, right? So it finds all of those relevant images, extracts all of those and provide that, and also summarizes those information for the user. Right. And this, I think pathologists actually find to be even more helpful because even if they do not trust the generation of the model itself, they often still would like to see how did their colleagues have discussed similar cases previously, right? And by retrieving the similar cases and summarizing that, that still provides very useful information to help the clinician. So there's a lot more information about this chatbot model that was recently published a few months ago in Nature Medicine. And we really view that this is a sort of like semantic search engine, right, for medicine, right, especially for pathology. Here, the users can type in text, right, what kind of things they're looking for, and the model then will retrieve relevant images. They can also just upload an image, and the model will also find similar cases before and summarize the text. And it's a publicly available search engine, and all of this is open source. So the last part I want to mention, now that we talked about the exciting applications of generative AI, I just want to end by also giving some caveats, right? So I think these AI tools can be very powerful, but there are also a lot of important questions that we need to answer, especially thinking about the potential biases in these generative AI models. So this is a paper that we published last year, where we basically studied different kinds of stereotypes and biases in these image generation models like DALL-E and Stable Diffusion. Right, so for example, if you take Stable Diffusion or DALL-E and you type, okay, I want the image of a software developer, here's basically the kinds of images that it would generate. Right? These are actual output from Stable Diffusion. And you can see that there's a really a strong kind of stereotype of what this AI model believes to be like a stereotypic software engineer. Right? In particular, right, there's only, I think, less than 1% of the generations of software developers are actually women. And similarly, right, these generative models also have a strong bias stereotype of what a poor person would look like, or a terrorist would look like, or a thug would look like. Right? So these are all example images that are generated by the model. We use these queries. Right. So in this previous paper, we systematically quantified that these current generative AI models, especially text-to-image models, do contain quite a bit of these stereotypes and biases, right? And I think this is still a really important open problem for the community to address and certainly to be aware of as we're using these models. So just to wrap up the discussion, right? So here we have discussed different areas of how we can actually use generative AI across the biomedical translation pipeline, right? For early stages, we've seen how we can actually use these tools as a way to help us to explore new spaces of molecules. We've seen the application of how to use this to generate new and to validate new antibiotics, right? Clinical trials, as we go down the medical translation pipeline, clinical trials is a key bottleneck, right? And we've seen examples of how we can actually use generative AI tools like Trial Pathfinder to help us to design the eligibility rules of clinical trials, right, to make these trials more inclusive and more efficient. Right. And lastly, we're also seeing how we can actually develop new generative AI models, like especially visual language chatbots, to help as assistants to pathologists, right, to clinicians. And throughout all of this, I think it's really important to think about how to evaluate and quantify and mitigate biases in these generative AI models. So here again are the references based on our works. And I want to especially acknowledge again my students at Stanford that led all of these works, right? Richard, Kyle, Zhi, and Federico, as well as our fantastic collaborators, especially Ryan Copen's team at Genentech, who's a close collaborator on these clinical trial part, and John Stokes from McMaster and his lab was a very close collaborator on this drug design side. So I'll stop here and happy to take any questions.
James, thank you so much. That was fascinating. Congrats to you and your team for really putting together a great program for, not only drug discovery or antibiotic discovery, but also coming up with the way to kind of create a bigger space in the clinical trial domain, and as well as some of the stuff you did with PLIP. I think that seems like a very powerful tool. One thing I just wanted to get your comments on is I noticed that you had created 70 different antibiotic candidates, and 58 got tested and proved to have some worthwhile to them, but there were like 12 that didn't work. Do you know why the 12 didn't work?
Question. So there's an apple that um, was not able to be synthesized at high yield, right? So basically, we sent these recipes to enemy in Ukraine, and they were not able to follow the recipes exactly on those 12 on those handful of the molecules. And there's a few reasons why that happens, right? Sometimes the algorithm, the AI model believes that a particular chemical reaction is compatible with certain building block fragments, right? But in reality, maybe those are hard to put together or are not very efficient in the lab, right? So I think that's one common reason why that did not work. Or it could also be that some of the building blocks may not have been available to enemy.
And I liked what you demonstrated with the ability to create the molecules and then go, create as many as you can resolve with your company, and then test them against known pathogens. And then you'll kind of know if you kill those off with your, which ones kill those off, you can identify which ones might be helpful. I'm curious if you can kind of do that process in reverse. Is there anything that you can know a priori about the pathogens and help guide the model creation, or the molecular creation of your antibiotic candidates, so to kind of like scale down and parameterize the search space?
That's a great question. And I think that is actually something we're actively working on, right? Because ideally, I view this as like a feedback loop, right? Where
Yeah.
we first generate a first batch of molecules, we test those against the real pathogens in the lab. Now we have additional data that can then go back and help us to guide the designs of even the next generation of new drugs, right? So the model becomes much more efficient going forward. So we're actually implementing that whole feedback loop. And you're absolutely right. I think there's a lot of actually information that the model does learn from the initial experiments that helps the model to explore more efficiently in the future.
And can you make predictions from those antibiotics, for example, which are successful? Can you get a sense of their the bacteria's resistance to it, or rather not immediate, obviously it kills it, but could you identify places where the bacteria might start to find resistance against it, and then somehow kind of head that off at the pass to extend the lifetime of the efficacy of that particular antibiotic?
So I think that's a really good idea, and I think that is the type of information that the model will be able to learn and leverage, right? So for example, when we train the current model, we actually give it a lot of existing data from previous drug screens against other pathogens, right? So the model does learn some information about what are the kinds of molecular motifs that are more likely to be effective against certain bacteria. Right? So it is bootstrapping off some of that prior knowledge and domain knowledge to help to guide the search space.
And once you discover these things, let's say you found that out of the ones that you created, you found that 6 were successful. Of those, they were able to kill off 4 of the 6 test pathogens that you gave to it. But let's say you find some that are particularly successful. Do you openly share the recipe with people, or is there some drug company that wants to snarp this up and go make a lot of money?
Yeah, so that's also a great question. So everything we did here is all open source, right? So we actually also made the AI model itself open source, as well as the products, right? So the discoveries of the AI model, right? And the reason why we did this is to really, I think, we want to basically help other people to basically use this algorithm as well to discover new drugs in their own space.
I think there's a broad moral, ethical kind of question as well, which I think actually, because you mentioned Alexander Fleming. And when he discovered penicillin, he gave it away because he felt that it was important for the world and that he didn't want it to become the purview of some foreign company that was just going to go and make a lot of money off it. So he gave it away. And there's some of the work that you're doing seems to be in that spirit, and then giving it away for free certainly doubles down on that kind of notion that this is stuff that should be shared with the world so that people's health can be protected and maintained, and that other people can then build on these ideas and make them stronger. So I was impressed with that kind of idea. And I also like your idea of kind of like looking to see how you can use the trial Pathfinder to actually kind of optimize your clinical trial work, so that you're not always looking at these Olympic athlete test subjects, but you're able to actually broaden things in a very informed way. And I'm curious if once you identified these kind of optimized sets of parameters, do you then route those through, say, human clinical trialists to see whether or not those look like they're reasonable, or that you can get, feedback from a human who's expert in clinical trial, or certainly domain experts, to see whether or not those things are actually doable?
Exactly. Yes. Yeah. So it's very much a collaboration between the AI model and the human clinical trial teams, right? So then we'll sift through all of those data from millions of EHR records, right? And then propose this data-driven design of the eligibility rules of the clinical trials. And then that would basically become one source of information that the clinical trial teams, or the human experts, would use to help them to guide the designs of the trials. So ultimately, the human teams in our studies, in our collaborations, they do evaluate, right, the designs of the AI model. And in these cases, we looked at, for example, in the lung cancer cases, the designs of the AI model is actually very reasonable to the humans, right? In particular, there are actually a lot of instances in the past where either by chance or anecdotally, where some of the actual human-led clinical trials end up looking very similar to the AI ones, right? Because there's a huge amount of heterogeneity that are in people, how people design these trials. And that's actually one source of information that we can use to further validate that the AI proposed designs are reasonable.
So in our last couple of minutes here, I'd like to, what you're presenting here are ways that I think are paradigm shifting for how people do drug discovery, and certainly things like antibiotic, looking at antibiotic efficacy and, the various properties of its success. You're almost touching on personalized levels of antibiotic kind of creation, right? You could almost tailor this, some of your trial Pathfinder related work. So you could actually dovetail those. And given some of the other things that you've optimized here, you're really trying to, I don't know, I get the sense that this is really kind of paradigm shifting for how you would train the incoming generation of people who do this type of research, whether it's drug discovery or whether it's clinical trials. How do you see these kinds of things influencing education? And are there any things that we need to be thinking about as we start thinking about, okay, in the next 20 years, this is what people are going to do when they do this type of work? Any thoughts on that?
Yeah, thank you for that. I think that's a really interesting question. We actually have sort of like a perspective paper coming out, thinking through like what should, for example, data science education look like, right? With all these generative AI tools like ChatGPT, right? And I think at the education level, I think there's a really tremendous opportunity, right, of how we can actually use these generative AI tools responsibly, right, as a part of the education, right? And certainly, I think we want to think through how can we teach our students and the future researchers of what are good ways of using language models and using ChatGPT to help them to assist their research, and also like maybe what are not so good ways, right, that they want to avoid. Right. But overall, I think one area where I think these models can be very useful for helping future researchers and in education and also in their own research going forward is really as a way to help them to generate new ideas. Right? Because these models have seen so much of the literature, right? Much more, they've read a lot more papers than we would have have time to ever possibly read. Right? So I think there are a good way of sort of summarizing literature and then try to identify gaps in literature and gaps in and propose new ideas that would potentially spark some creative ideas for the human researchers.
Well, James, thank you so much for sharing this with us. This was really inspiring, and this is all absolutely a mountainous amount of work. So, congratulations to you and to your team, and thank you for
Thank you
sharing
very
it.
much. Yeah.
Yeah, and thank you everybody who is watching or will soon watch us on YouTube, and everyone have a